Welcome to Creative Diagnostics channel. This video we are going to talk about Toxoid vaccine in Subunit vaccines platforms. We will talk about the following 4 aspects. A vaccine is a biological formulation designed to give rise to a protective and ideally long-lasting immune response against a pathogen without causing disease. Vaccines can be either prophylactic or therapeutic, with prophylactic vaccines given before encountering the pathogen and therapeutic vaccines given after encountering the pathogen. Many different approaches have been employed in developing vaccines. For example, live attenuated vaccines consist of live pathogens from bacteria or viruses that have been weakened by heat or chemical methods. Recombinant subunit vaccines consist of recombinantly produced subunits of a pathogen that stimulate the host immune system and contain only a portion of the pathogen. Some pathogens produce exotoxins that contribute to or cause disease, such as tetanus toxin, botulinum toxin, and diphtheria toxin. Toxoid vaccines, consisting of chemically inactivated toxins mixed with adjuvants, produce a protective immune response. Toxoid vaccines have been used since the 1920s and are known to elicit a strong and protective humoral immune response after initial vaccination, but several booster shots are required to develop longer-term immunity. Unlike a vaccine against an entire organism or virus particle, which contains multiple immunogenic epitopes including antigens derived from surface protein epitopes, viral capsids, bacterial cell walls, glycoepitopes, and polysaccharide epitopes, a toxoid vaccine contains a single protein. However, within the single protein, multiple antigenic epitopes are present, some of which are more immunogenic than others. The process of turning a biologically active toxin into a toxoid usually involves prolonged chemical treatment with formalin to inactivate the toxin. When the toxoid vaccine is injected into the body, it is recognized as foreign and results in a strong humoral immune response, producing antibodies directed against specific antigenic epitopes preserved on the toxoid structure. With repeated boosts, the immune system develops a moderate humoral memory response lasting for several years, allowing for a rapid and robust response with future exposure. However, repeated boosts are required periodically to maintain lifetime protection. When a toxoid vaccine is administered, the antigen-presenting cells, APCs, such as dendritic cells generally process the toxoid and present peptide antigens on their cell surface using major histocompatibility complex class II, MHC2 molecules. These MHC2 peptide complexes act as a signal for T cells, specifically CD4+ T cells, helper T cells, and trigger a cascade of immune responses, resulting in the activation of B cells and cytokine secretion in the host body, ultimately leading to differentiation of B cells to antibody-producing plasma cells and memory B cells. The principles of toxoid vaccine production and action can be seen in the schematic diagram. Firstly, the pathogenic bacteria secrete toxins that contribute to disease. Toxins are inactivated or attenuated using heat or formalin, resulting in creation of toxoids. Then the toxoids are absorbed with adjuvants to increase immune response. The host body generates antibodies in response to toxoid, providing immunity against the toxin. Classic examples of toxoid vaccines are tetanus and diphtheria vaccines, which use chemically inactivated toxoid combined with an adjuvant such as alum to effectively protect against the disease. The diphtheria, tetanus, pertussis vaccine was one of the first vaccines used in humans. It protects against 3 serious and potentially fatal diseases that affect children and adults. Diphtheria is an infectious, potentially fatal disease caused by Corynebacteria species able to produce highly potent diphtheria toxins. Tetanus is an infectious, potentially fatal disease caused by anaerobic bacteria, Clostridium tetani. Pertussis is a severe, infectious, and highly contagious respiratory disease caused by Bordetella pertussis. Prior to the first registration of the DPT vaccine, vaccines containing each component were developed separately. First successful approach to active diphtheria immunization was the mixture of toxin and antitoxin. In 1920, Ramon showed that the diphtheria toxin, after treatment with heat and formalin, lost its toxicity while maintaining immunogenicity properties. As a result, toxin, antitoxin was replaced by diphtheria toxoid. In 1924, a chemically inactivated tetanus toxin was developed and is used for active immunization against Clostridium tetani infections. It is now known as tetanus toxoid. Initially, tetanus toxin was inactivated using iodine trichloride. Now formaldehyde is used. Whole cell pertussis vaccines are suspensions of inactivated B pertussis cells. In 1948, they became available in combination with diphtheria and tetanus toxoids as the diphtheria whole cell pertussis and tetanus, DTWP vaccine. Current pertussis vaccines are based on various combinations of the 5 main pertussis antigens, including filamentous hemagglutinin, FHA, pertussis toxin, PT, pertactin, PRN, and 2 Fimbria proteins, FIM2 and FIM3. The DTaP vaccine has fewer side effects and is better tolerated. DTaP vaccines have been produced all over the world. The production of toxoid vaccines begins with the use of suitable bacterial strains capable of producing large amounts of toxin in fermenter cultures, followed by toxin collection, filtration, sterilization, and chemothermal detoxification. The native toxoid is purified, concentrated, salted, sterilized by filtration, and finally adsorbed on the adjuvant, most frequently on aluminum salt, usually aluminum hydroxide or aluminum phosphate. At present, various combinations of the DTaP vaccine have been licensed. For example, with HIB component, inactivated poliovirus component, or hepatitis B component. DTaP vaccines were initially used as toddler and school entry booster vaccinations, then several years later were introduced for primary vaccination. According to the recommendations, all adolescents should receive the DTaP vaccine as a booster dose. Currently, in many countries, the primary schedule for tetanus, diphtheria, and pertussis vaccination includes 5 doses of vaccine given in the first 2, for 6 and 15 to 18 months of age, with the next dose at the age of 6 years. The DTaP vaccination is recommended for pregnant women to protect newborn babies against pertussis before administration of the first dose of the DTaP vaccine. Studies of DTaP vaccination during pregnancy showed that an infant benefits from high levels of transplacentally derived material antibody, and vaccination during pregnancy provides protection during the greatest risk period, before infant vaccination begins. We need to improve existing vaccines or develop new ones to induce effective mucosal defenses for early life and long-term protection for infants, adults, and the elderly in response to changing epidemiological conditions. Anti-diphtheria antibody level decreases with time, and without booster vaccination, adults may again become susceptible to diphtheria. Invasive non-toxigenic C. diphtheria strains infections are now occurring. Need for a new vaccine that would be effective in the prevention of not only diphtheria but also infections due to non-toxigenic C. diphtheria strains that do not meet the criteria of diphtheria. Investigators attempted to use a novel system to develop oral tetanus toxoid and induce a mucosal immune response. New adjuvants for C. tetani toxoid have been invented with a reduction in size to nanoparticles, which enhances the immune response. Typhoid fever, an invasive bacterial infection caused by Salmonella enterica cerevar typhi, remains an important public health threat worldwide. There is no very effective protective vaccine against typhoid. The 2 typhoid vaccines currently recommended by the World Health Organization are an oral live attenuated vaccine and an injectable subunit vi-capsular polysaccharide vaccine. Several groups have developed typhoid conjugate vaccines using vi-polysaccharide from different sources and using different carrier proteins for conjugation. The introduction of typhoid conjugate vaccine into routine immunization is considered to be one of the most effective interventions for the youngest age groups. BONTs, botulinum neurotoxins, are the most potent protein toxins known to humans to date. They are 150 keta protein toxins that are proteolytically processed to form a 100 keta heavy chain, HC, and a 50 keta light chain, LC, linked via a disulfide bond. BONT LCs are released directly into the cytosol of the motor neurons, inhibiting neurotransmitter release by the motor neurons. This leads to long-lasting flaccid paralysis. Once inside a neuron, BONT LC can stay active for a prolonged time of days to several months depending on the BONT serotype. Therefore, a vaccine to BONTs has to be highly efficient in removing the toxins from the circulation before neuronal cell entry. No cure exists for botulism, but protective vaccination with botulinum neurotoxin toxoid, prepared by formalin inactivation similar to TENT and DT toxoids, has been shown to be effective in preventing botulism. Protective vaccination with botulinum neurotoxin toxoid, prepared by formalin inactivation, has been shown to be effective in preventing botulism. However, the development of a toxoid vaccine against BONT presents many challenges. Recombinant protein or subunit vaccines do not contain whole organisms and rely on host immune responses against identified immunodominant antigens from bacteria, viruses, or toxins. Toxoid vaccines are comparatively favorable in terms of production, transportation, and storage. The vaccine candidate can be lyophilized to powder form, has a better shelf life, and can be transported and stored at room temperature as well. On the other hand, Toxoid vaccines are more costly to develop and usually require the addition of adjuvant as part of vaccine formulation to achieve effective immune responses, and inducing a protective T-cell response and immune memory in addition to the humoral response is challenging. All toxoid vaccines share the necessity of booster shots at regular intervals to generate a strong and effective immune response, and particularly to create immune memory. Utilizing specific mutations in the toxin gene to knock out toxicity, as is currently being explored for recombinant toxoid vaccines, would mitigate the risk of active toxin production, but still a risk of DNA incorporation and reversion remains. Overall, to improve existing vaccines, new strategies are required to address unmet needs, particularly for pathogens with antigenic hypervariability or pathogens with multiple serotypes, and for pathogenic exotoxins. Thank you for watching. If you want to gain more knowledge, subscribe to the Creative Diagnostics channel. We are dedicated to help you succeed and advance your research progress. Visit our website for more information.